Risk factors for cancer trigger the mutation. We call those risk factors carcinogens. Carcinogens can result in mutations in the DNA to the point that the cells neither look like nor behave like normal cells. Carcinogens can cause cells to take on malignant characteristics. Look at that word carcinogen. Genesis. Something is capable of generating a change in the DNA that results in a malignant condition. Carcinogens result in mutations of genes that result in malignant cells that neither look like nor act like normal cells. A carcinogen is capable of mutating the DNA to the point that a malignancy can occur. So a carcinogen initiates the DNA damage. There's some early cellular change that's potentially reversible, but if it's not, that change is promoted and allowed to get worse and worse. The damaged DNA progresses to the point that the person has a malignant neoplasm. Initiation, promotion, and progression. We are now going to identify some prototype carcinogens, not all of them, but prototypes. We are going to discuss the process whereby carcinogens can result in malignant cellular change. And we're going to discuss the concept of genetic mutation as it relates to cancer. Common carcinogens. Damaged DNA can be related to radiation, ultraviolet light, environmental pollutants, smoking, asbestos, viruses, and other carcinogens that increase the risk for DNA mutation, which can ultimately result in cancer. DNA can be accidentally damaged. Accidental DNA mutation. When the DNA replicates itself, the errors in the replication are not repaired. The cells produced by that replication error can be either benign or malignant. Replication errors occur most commonly in epithelial cells, cells that are rapidly turning over. But these accidental DNA mutations are often in areas not likely to result in cancer. DNA can be damaged related to chronic inflammation. For example, visceral fat, the fat that you can't see, the fat inside the abdominal cavity. That visceral fat results in chronic inflammation, and that can be a carcinogen capable of producing a malignant cell. Inflammatory bowel disease, chronic inflammation, colon carcinoma, chronic ulcerative colitis, Crohn's disease, which we'll talk more about with GI, but those are triggers for malignancy related to chronic inflammation. Damaged DNA related to chronic inflammation, reflux esophagitis or GERD, gastroesophageal reflux disease, that's chronic, can cause inflammation in the esophagus, resulting in adenocarcinoma of the esophagus. Hepatitis would be another example. Chronic inflammation in the liver, resulting in liver adenocarcinoma. We'll talk more about that when we talk about viral hepatitis. And also we'll talk more about GERD in the module on upper and lower GI disorders. Hormones may drive cells to malignant changes or promote growth of malignant cells. For example, estrogen may promote the growth and the development of cancer of the breast and in the endometrium. Insulin is an anabolic hormone, which can promote the growth of certain malignancies. Immunologic mechanisms are also risk factors for cancer. And by immunologic mechanisms, I'm talking about reduced immune surveillance, inability of the immune system to recognize an antigen on a cancer cell and attack it, defective cytotoxic T cells. Click on this video and you'll see how the immune system is supposed to work. And as you look at the immune system attack bacteria and viruses, think about the immune system also attacking malignant cells. Chemical carcinogens, such as cigarettes, result in cell mutation that can result in malignant DNA mutations. Chemical carcinogens can alter cell enzyme synthesis. Chemical carcinogens can alter cell replication and interfere with cell regulation, resulting in uncontrolled cell division. So mutating the DNA is what causes cancer. Uncontrolled cell division is a characteristic of malignant cells. Chemical carcinogens usually have a cumulative and time delay effect, meaning that it can take years following exposure to a carcinogen before you mutate the DNA to the point that you have a malignancy. That helps to explain why certain malignancies are more common as a person ages. Cumulative damage in the DNA has resulted in a malignant DNA change. I think it's important to know that there are no safe UV rays. Exposure to UV rays over a period of years results in cumulative damage to your DNA to the point that the DNA mutates and a person can develop a variety of skin cancers. We're learning more about viruses every year. There are oncogenic viruses, viruses capable of mutating the DNA, resulting in cancer. Two prototype examples would be human papillomavirus HPV 1618 and cervical cancer. All cervical cancer is the result of viral DNA mutation. There are other viruses other than 1618 that we will talk about with reproductive disorders. And then hepatitis B and C, which are viral. That persistent virus, that persistent inflammation can mutate the cells, mutate the DNA, with resulting hepatocellular cancer. A virus sends a malignant message. The genome of the virus gets inserted into the host DNA. And when the virus inserts into the host cell DNA segment, there's a malignant message, and the cell takes on malignant characteristics. It neither looks like nor performs like a normal cell. Let's discuss how free radical damage could result in malignancy. A free radical is an atom that is missing an electron in the outer shell. So sun, UV rays, viruses, cigarette smoke, and other carcinogens can bump electrons out of the outer shell. That free radical is unstable, so it goes to an atom next to it and steals an electron, which makes that atom a free radical, and so on until there's so much free radical damage done to the cells that it becomes vulnerable to DNA mutation from the outside. Outside forces can mutate the DNA because the outer shell or the cell membrane is so unstable that the cell cannot be protected. Protein starts out as a chain of amino acids. That chain of amino acid folds to form a normal protein, or it unfolds or misfolds and becomes a trigger for malignancy. An abnormal protein inside a cell can change the message that the cell receives, resulting in a malignancy. There are genetic factors that predispose a person to malignancy. Cancer occurs in 10% of persons with one affected first-degree relative. Heredity, a risk factor for cancer. Brca mutation. Brca is a healthy tumor suppressor gene. We all have it. A healthy Brca tumor suppressor gene helps protect us from cancer. But if there's mutation of that tumor suppressor gene, there's failure of DNA processes, which increases the risk for breast and other cancers. Many cancers have been linked to obesity. So let's discuss the link between obesity and cancer. And by obesity, we're primarily talking about visceral fat, the fat inside the abdominal cavity. And we know that obesity means body mass index of 30 or above. One of the things that links obesity to cancer is estrogen, postmenopausal estrogen levels. As a woman gets older, estrogen levels fall. But when you're obese, estrogen continues to be synthesized in adipose or fat tissue. That leads to estrogen levels that are higher than you would expect for the age of the woman. So that increased estrogen level found in obese women increases the risk for breast and endometrial cancer. Obesity in men is also an issue when it comes to breast cancer in men. Individuals who are overweight or obese often require more insulin to move glucose into the cell. And those higher levels of insulin are a risk factor for malignancy. Insulin is anabolic, and some malignancies have insulin-like growth factors, meaning that they will respond to high levels of insulin. For example, you'll find more polyps in the overweight and in the obese because of insulin-like growth factors promoting the development of polyps. And once an individual has a polyp, continued DNA mutation increases the risk for colorectal cancer. So it's interesting to know that obesity and inflammation as a risk factor for cancer may be responsible for 1/3 of all cancers. 1/3 of all cancers may be related to obesity. And once an individual has certain malignancies, overweight and obesity contributes to recurrence. On this slide, notice the normal cell has undergone uncontrolled cell division, and the cells that are produced as a result of that DNA mutation neither look like nor perform like normal cells. Uncontrolled cell division is a characteristic of malignant conditions. Now let's look at the question. What causes cancer? What causes malignant cellular change? Some trigger, which we call a carcinogen, that trigger results in DNA mutation. DNA mutation causes cancer. Look at the normal cell and then look at the malignant cell. Cancer, cells that neither look like nor perform like normal cells. Uncontrolled cell division, producing cells that neither look like nor perform like normal cells. We've addressed this objective briefly before, but let's describe which cells are likely to be most vulnerable to DNA mutation. Cells are most vulnerable to DNA mutation during periods of cell division. So cancer arises most often from cells undergoing frequent mitosis, and that would be epithelial cells, which is why most malignancies, or many malignancies, are carcinoma. Carcinoma means an epithelial malignancy. Cancer arises most often from epithelial cells because they are so rapidly dividing. Notice that there are usually 3 replication errors made per cell cycle. But we have tumor suppressor genes, and when they are healthy, these errors are repaired. Tumor suppressor genes help promote DNA repair processes so that the cells that are produced during the cell division process, that those cells are healthy. When you have mutated tumor suppressor genes, then those errors are not repaired, and that helps promote the development of malignancy. Look at the review question. What are you most likely to see on the pathology report of a person who has cancer? We can eliminate C and D immediately. A lipoma is not cancer. A lipoma is a benign tumor involving fat. Dysplasia is not cancer. Dysplasia is an abnormal volume of tissue. So what are you most likely to see? Sarcoma, which is a connective tissue cancer, or carcinoma, which is an epithelial cancer. Carcinoma. Cells are most vulnerable to DNA mutation when they are actively dividing, and epithelial cells are highly mitotically active. Let's talk a little more about how DNA mutation can alter the cell repair processes, which allows normal cells to become malignant. DNA mutation. Remember, replication errors should be fixed by tumor suppressor genes. But what if they're not? Let's first look at how normal cells handle DNA replication errors. Tumor suppressor genes protect us from DNA replication errors. If damaged DNA cannot be repaired, abnormal cells are not allowed to undergo mitosis. If damaged DNA cannot be repaired, abnormal cells are kicked out of the cell cycle. If damaged DNA cannot be repaired, abnormal cells are told to commit cellular suicide, to undergo apoptosis. Normal cells respond to external signals, like number 1 on this diagram. They respond to external signals, which helps control cell division. 2, there's repair of damaged DNA. 3, there are alert signals when something goes wrong. 4, cells stay within boundaries. And 5, cells commit apoptosis when needed. This cell says, I passed the test. It passed the 3 checkpoints. The cell has grown large enough to divide. If not, it will be kicked out of the cell cycle. The DNA has correctly duplicated, or if it hasn't, duplication errors will be repaired. The cell has proteins needed to separate the chromosomes. If all these things are healthy, in the normal cell, the cell will engage in mitosis. It's so important that healthy cells repair damaged DNA. Remember, there's 3 replication errors per cycle. But healthy cells will repair damaged DNA, which reduces the risk for cancer. P53 is a tumor suppressor gene that's very important. P53 allows DNA to repair itself. P53, a healthy tumor suppressor gene, takes damaged cells and kicks them out of the cell cycle. Or P53 will tell damaged cells to commit cellular suicide, apoptosis. When P53 mutates, damaged DNA is not repaired, damaged cells are not kicked out of the cell cycle, and damaged cells do not commit apoptosis. So let's summarize this again. Cancer. There's a tumor suppressor gene mutation, so there's no repair of damaged DNA. There's no stopping of damaged DNA. When tumor suppressor genes are mutated, there are no alert signals sent out when something goes wrong. What if there is no alert when something goes wrong? Cells do not commit cellular suicide. Damaged cells do not undergo apoptosis. They're allowed to reproduce. And every time they divide, they can become more and more and more abnormal until they become malignant. And even once they become malignant, they continue to become more and more and more abnormal. Carcinogens can cause DNA replication errors, but we want to repair those replication errors. Again, let's think about P53 and the function. A healthy P53, a healthy tumor suppressor gene, will slow down or control cell division, will repair DNA mistakes, or tell cells when to die, when to commit cellular suicide or apoptosis. I think it's interesting to know that more than 1/3 of cancers have a mutated P53. P53 is one of the tumor suppressor genes. More than 1/3 of cancers have a mutated P53. Click on this website and look at protein misfolding when the amino acids misfold. Brca1 is a tumor suppressor gene, and when the amino acids misfold, that can be an etiology for malignancy. You could kind of summarize it this way. Tumor suppressor gene mutation. When tumor suppression is turned off, the oncogene is turned on. When tumor suppression is off, the oncogene, or the gene capable of causing cancer, is on. Healthy tumor suppressor genes put the brakes on cell division. When it's mutated, it's like pushing the accelerator down, uncontrolled cell division. Healthy tumor suppressor genes repair damaged DNA, code for apoptosis, and kick damaged cells out of the cell cycle. Failure to do these things increases the risk for cancer. The trigger or risk factor initiates DNA damage. That trigger or risk factor is called a carcinogen. If there's a mutation in the tumor suppressor gene, then damaged DNA is not recognized and repaired. That mutated p53, a mutated tumor suppressor gene, does not recognize and repair damaged DNA. In addition, abnormal cells are not kicked out of the cell cycle, and cells are not coded for apoptosis. The DNA damage continues to be promoted and get worse. More and more DNA mutation will eventually result in a malignant change. How would you answer this review question?
What has to happen before a cell can become malignant? Does it need an exposure to a virus, DNA mutation, genetic tendencies, or immune suppression? Hang in there, we're almost ready for a break. And the answer is DNA mutation.
In order for a cell to become malignant, the DNA has to mutate. Now, exposure to a virus may cause the mutation. Genetic tendencies may result in DNA mutations. And failure of immune suppression may result in failure to recognize abnormal cells and destroy them. But for a cell to become malignant, the DNA mutation has to occur.
And you can find a summary of the characteristics of malignancy on page 96 of your text. All right, let's take a break and we will be back for part 3.